Hello, my name is Professor Matthew Schmidt, and I'd like to welcome you to Genetics. In today's session, we're going to start talking about what really should be called molecular genetics. We're going to start at the beginning, as you would expect, with DNA structure and replication, with the idea that DNA is the genetic material. Now, I suspect you're saying, yes, we know that, but most genetics courses are taught in what's usually referred to as an historical fashion. In other words, most people, even who are not biology students today, know about DNA, but Mendel didn't know about it, and Sutton and Morgan didn't know about it. So, in other words, all of the genetics we've done so far, and we've done quite a bit, it was all done and can be fully understood without knowing anything about DNA or the chemical nature of the gene. So now we're ready to, in essence, to say if we were going forth in historical time, and it was about 1925, we're at a point where we're ready to begin to explore what's going on at the molecular level. Depending on your course, it may or may not go into this part so much. We're going to look at some experiments that help to elucidate the fact that DNA is in fact the genetic material. I suspect most courses do expect you to know this, so if not, I guess you don't have to. So basically, this is a little bit of a recap of some of the things I just said. Structure and function are so intimately intertwined, not just in biology, but particularly in biology. So we've learned a lot about the function. In other words, we've learned how to predict the results of crosses, and we know how to map genes. Really, ultimately, we do have to relate that to the structure of what the genetic material really is. So, as I said, up until now, everything we've talked about has not taken into account this chemical nature. Another thing, just to keep in the back of your head, is that it's also not taken into account how genotype determines phenotype. In other words, it's all well and good for Mendel to have said, okay, well, Big P makes purple flowers. I'm not criticizing him, but he had no idea what Big P was, and he had no idea, certainly how whatever it was translated the purpleness into reality. So these are all questions we're going to discuss as time goes by. As I say, it's time to find out and get to the bottom of all this. So before put yourself in the position of a scientist who's thinking about this. What are the requirements if we're looking for a molecule for the genetic material? Well, based on everything we know, it has to be able to store and express information. That's probably obvious, but what I mean by that is there are lots of ways to store information, but we didn't discuss this. But take a molecule like starch. It's just a repeating of glucose over and over and over and over again. There's really not a lot of potential for information storage in that type of molecule. So we have to be able to envision a system. Think of if you were looking at a Blu-ray DVD. It's a digital system, right? Zeros and ones are the code that we're using, the symbols that we're using. So there are loads of ways to store information, but we need to find a molecule that can do it. Furthermore, it has to be able to replicate itself. Remember, it was known that how the chromosomes behaved during mitosis and meiosis, and it was very clear that whatever molecule this was, a copy has to be made of it. So it has to be able to make that copy. And this is somewhat redundant with what's already been said, but there has to be a lot of variation. And what I mean by that is in the molecule, we have to be able to account for not only the fact that there's variation within a species at particular traits and even amongst different traits in that species. But since we've seen that fundamentally genetic processes work similarly, whether it's a pea or a fruit fly or whatever organism, a mouse, ultimately we have to be able to account for all of the genetic variation in the biological world. So there has to be a ridiculous capacity for differences in the information that's stored. And I only mention mutation here just because, we're going to have big talks about mutation. But mutation just means a change, and it was known even before it was understood what DNA was that changes could occur. Remember we said that the white-eyed allele in the fruit fly was a mutation of the normal wild-type red-eyed allele. So, in other words, it was known that these changes could take place. We have to choose a molecule that has a capacity for this type of change. Okay? So when I ask here, what do we know so far? I don't mean us, you and me. I mean in about 1925, what was known given Mendel and Sutton and some of the early fly work, etc. So one thing Mendel said, right, was that genes are units of information. So that's for sure. We further know that they are physically located on chromosomes. In fact, you could say that those are the two major, we had talked about Mendel and chromosomes fusing together conceptually, allowing us to do things like make maps of genes and understand what those maps really mean. Something we haven't talked about but was known is that biochemists had examined chromosomes and sort of in a bulk way determined that they were made of proteins and DNA. So DNA was known about. It just wasn't thought to be particularly important, which sounds strange now, but it was found in every chromosome and it was really thought to be part of like a scaffolding material. As far as anyone knew, proteins did, being enzymes, they did all the work. And, well, I guess I'm getting ahead of myself, but since it's known that the genes were on chromosomes, right? And the chromosomes were made up about half DNA and half protein, it was really widely assumed in the early days that protein was the genetic material. The reason is because DNA, as we're going to see, but it only has 4 different bases that can create the variability, whereas proteins have 20 different amino acids. So in theory, you can have a lot more variability with protein, but just remember that DVD. There's only 2 symbols there that can encode the whole 5 hours of some epic movie in high definition. So you really don't need a lot of subunits or symbols in a code in order to encode a lot of information. But it was assumed that proteins were the thing. There was a lot of variety of proteins. It was incorrect, but it was thought in the early days that DNA might just be a repeating polymer, almost like starch. And the fact that proteins were catalytic, a lot of people thought they described the genetic material almost as like a super enzyme. Somehow it was, it may sound odd now, but that's all biochemists were into protein in those days. So, turns out they were wrong, but how did we find that out? So it all started with an experiment, performed by a gentleman named Frederick Griffith. He was a microbiologist studying infection. So Griffith's experiment is usually thought of as the first indication that DNA was the genetic material. Although you should be clear that he didn't actually say that. You'll see as we go through it. Because really what he was doing, he was working with bacterial pneumonia using mice as a model organism, and he performed what's now a famous experiment that gave a clue when people thought about it later as to what the nature of the gene really was. So let's just look at what he was doing. He really wasn't a geneticist. He was trying to figure out various things about infection using this pneumonia. So first of all, he used a bacteria called Streptococcus pneumoniae. It is one that often still today causes pneumonia. Other versions of Streptococcus cause like strep throat. So it's a fairly common pathogen as well as a normal member of the bacteria in most people. But in the case of Griffith, he had identified 2 strains of this bacteria. Strain is sort of like a subspecies, a particular line of the bacterium that has certain characteristics. There was the S strain and there was the R strain. And he named it S because when grown right on a plate, you could see that it was smooth and shiny. I forget if the S is for smooth. Yeah, for smooth. But in other words, he could tell the difference just by looking at it, not even under a microscope. It also turned out, so the R was rough, in other words, not smooth. You could tell the difference. But the more important thing was that the S strain was very virulent, that is, very able to cause disease, and it would cause pneumonia and often death in mice that were infected by it. But the rough strain never caused illness. So he was trying to figure out what the difference between them was and just some general things about infection. We now know today that the S strain both look smooth and cause disease because they're surrounded by a structure called a capsule. If you ever take microbiology, you might talk more about that. But the capsule is able to cause the virulence. What it really does is that it inhibits the host defense system from being able to deal with that bacterium. But getting a little off into microbiology here. So he's got these two strains, and he did a famous experiment that we've sort of pictorially illustrated here. There are a couple of steps in it. Now, he didn't necessarily do these one after the other, but we're looking at his data conceptually to try and figure out what it meant. So step one here, what he observed is this. If you take the S strain bacteria and literally take it up, put it into the mice get bacteria. They get bacteria. Excuse me, they get pneumonia from the bacteria, and they get sick and die. So this upside down mouse here is obviously a dead one, right? Also, he didn't really know it at the beginning, but the S strain we're just showing here, this is Streptococcus. They go together in chains like that. But also what's surrounding them is the capsule, which is why the S strain is virulent. Okay, but basically the bottom line of this part is if living S cells are injected into the mouse, it gets sick. If you do the sort of similar experiment with the rough strain and inject that, the mouse is okay. It's fine. So the bottom line at this point, and you can see here that these bacteria don't have the capsule, which is really the reason why. So you got this S strain with a capsule that causes illness. You get this R strain without a capsule that doesn't cause illness. So far, it's not that complicated. Now it gets a little bit more interesting, because what he did was he took the S strain and boiled them, which kills them if you boil bacteria long enough, most of them anyway. And it may sound like an odd experiment, but when he was convinced that the S strain cells were dead, he took them and injected the dead cells into the mouse, and nothing happened. So these are supposed to look dead up here. They're sort of flattened out and messed up. But you might be saying, well, yeah, of course it doesn't do anything. But he wasn't 100% sure that you needed living cells for an infection. In theory, there could have been some other explanation. But this is important for our purposes. So dead cells do not cause an infection. That's the bottom line of this part. But the most interesting part, and the part that serves us genetically the most, is the next step, the last important step here, is he did this experiment where he took the dead S strain bacteria, like the ones we were just talking about, and he mixed them together, literally physically mixed them, with live R strain. So here we have dead S plus live R, right? Now remember, neither of those on its own was able to cause infection, right? You would think, therefore, that together they couldn't cause infection either. But if you literally put them together, let them sort of mix around and percolate for a little while, I think he was surprised. He put the mixture right into the mice, and they did become ill, get pneumonia, and probably die. So what's going on here? What the heck? Well, it turns out that when he thought about the results, I'm sorry, another very important thing just to complete the circle of this experiment is after the mice became ill and died, he cultured them for bacteria to see what it was that killed them, and he very importantly recovered growing, living S strain bacteria, smooth ones. So they were smooth. What this very, very strongly implies is that it sounds a bit odd, but that somehow, even though the S cells were dead, they were somehow able to teach the R cells how to become S cells. In other words, how to make a capsule. And that requires information being transferred, right? So Griffiths basically said that some information from the S cells, even though they were dead, was able to transform the R cells so that they became virulent and they became smooth. And we can also say, really, what was happening was they learned how to grow a capsule, if you want to think of it that way. Now, Griffiths referred to whatever the information was as the transforming factor. He, like everyone else in those days, believed it probably was a protein and really was not able to characterize it much further. But information is what genetics is about, you guys, right? So the reason we're highlighting this is because the first time we've seen information in the form of some chemical, it had to be, right? The dead cells were dead, so something in them had the information about making a capsule, and it was able to be transferred into the R cells and utilized. So we still use this term transformation in genetics for this type of phenomenon. We now know that DNA is the thing that's being uptaken by, one cell from another. But so Griffiths didn't say DNA was the transforming factor. He just said there's some transforming factor here. Now, approximately 15 years later, Avery, McLeod, and McCarty were working in Rockefeller University, and they published a paper in 1944 that was pretty definite about the fact that DNA was the transforming factor, and what that really means is that genes are made of DNA. That they basically repeated Griffith's experiment. It's a little bit technical what they did, but there's a couple of ways you can do it. They said, look, we want to know what the transforming factor is. Technology had advanced a bit since 15 years before. So one thing you can do, or they did, was to take the whole mixture and to fractionate it. Usually this is done by centrifugation, but basically carbohydrates, lipids, proteins, and nucleic acids, including DNA, are four of the major types of biomolecules in any cell, right? And they have different characteristics and weights, etc. So you can separate them from one another. And they tested each fraction for its ability to transform, as we just saw. And it turned out that only the DNA fraction was able to transform. So this is first real evidence not of the transforming factor, but that DNA is the thing that's doing it, and that therefore DNA is the genetic material. Now, a couple things. Another thing that they did, just so you can, in case it's emphasized to you, they took the mixture and they treated it, for example, with a protease, an enzyme that destroys proteins. And there are also enzymes that are DNases that destroy DNA. So in the presence of proteases, it still worked. That implied very strongly that it wasn't a protein that was doing it, right? But in the presence of DNases, it didn't work anymore because the DNA had been ruined. But it's hard to, relate to in our day and age. But at the time, there was a lot of, let's just say, this was not a very easily accepted idea. A lot of people were wary about the experiments, how they were done. And I think people just really had it in their head that protein was the thing, and they didn't feel comfortable with DNA being the genetic material. Well, that was just too bad, because further confirmation came in 1952. In the meantime, certainly people were continuing along these lines and doing the experiments and getting a little bit more comfortable with the idea that DNA was the genetic material. But this next experiment is usually considered the one that really put to rest any doubt, and it's called the Hershey-Chase experiment. Well, this was done in as I said, 1952, and they provided evidence that was almost indisputable that it was DNA and not protein. Really, this is what they were focusing on, that DNA was the genetic material. They used a totally different system and different methods, so it's good when you get independent verification from another whole system, right? It shows that it's not just something going on with mice and bacteria. They worked with bacteriophages. Many of you are familiar with that term, but these are viruses that infect bacteria, and it literally means bacteria eaters. They don't really eat them, but before they knew what viruses really were, that's what they called them. Bacteriophages are interesting because they are made up exclusively of protein plus DNA. And furthermore, when a virus infects a cell and sort of takes it over, it's very clear that some information from the virus is entering into the cell. So Hershey and Chase reasoned, well, let's find out. Is it the protein part of the virus that goes into the cell, or is it the DNA part that goes into the cell? I guess it could have been both, who knows, but this was the basis of what they were trying to accomplish. So let's look at their experiment in a little more detail. Before we can fully understand that, obviously this is sort of in retrospect, I'm going to tell you quickly how a bacteriophage works, but you need to understand this experiment. They had to figure it out, which is, hard. We just have to interpret it. So here is a bacteriophage. It actually looks like a little spaceship or something, and while this is a diagram, it really does pretty much look like this. If you look at a picture through an electron microscope, it's fairly amazing. But the bottom line is, always there's DNA, which is inside of the head here, and all of the rest of this stuff, this to the landing gear, all of it's proteins. So the body, if you will, of the virus is made of protein, and DNA is housed inside of there. So, we now know that the DNA is, I'm getting ahead of the experiment, but when this virus, if you want to think of it, lands on the outside of a bacterial cell, what it does is it just injects the DNA right into the cell. The whole point of the protein part is that that's a vehicle that moves the DNA from one cell to another, right? So once it's brought it there, it's fulfilled its function. It's done. So it turns out that this is true, that only the DNA goes in. But let's see how that they determined that was what was going on. So just looking over here, there's DNA surrounded by protein. It's got to transfer information into the cell. As far as Hershey and Chase knew before they did the experiment, they didn't know what it was. I'm telling you that it's DNA, but let's see what they did to find that out. So there's a lot on this slide, I apologize, but it's basically, here's the idea, and it's a beautiful experiment. They grew one group of bacteriophages on media that contained radioactive phosphorus. It's called P32, which is a heavy version of phosphorus. Now, it's sort of luck, but it's very important that you understand that DNA has phosphorus in it, and proteins do not have phosphorus in it. Conversely, proteins, I hope I said that just right, DNA has phosphorus in it, proteins don't. Proteins do, however, have sulfur in them, and DNA never has sulfur. So the way that they were trying to tell these apart was they grew one bunch of phages on radioactive phosphorus. If that happens, the DNA will become labeled, in other words, detectable, because it's radioactive now. If you grow them on radioactive sulfur, which is S35, a heavy version of sulfur, only the protein will become labeled or hot. Sometimes they call it hot if it's radioactive. All right? So you've got these strains, keeping them separate. One where you can sort of keep track of the protein because you grew it on radioactive sulfur, and one where you can keep track of the DNA because you grew it on radioactive phosphorus. And then separately, they infected bacteria with each of the strain. Now, they literally let the bacteriophage percolate and interact with the bacteria for however long, 10 or 15 minutes, and they put the mixture in blenders. It's sort of funny because it wasn't any big fancy piece of equipment. It was literally a Waring Osterizer blender, they called it. It was a kitchen appliance that I think they brought in from home. In fact, Oster is still a brand of blenders, but the point is it's nothing fancy. The blender, the idea of it was they just wanted to shake everything up really good so that whatever was not stably incorporated inside of the cell would sort of just fall off, literally. So they centrifuged the mixture then, the idea being that the cells, the bacteria, will fall down to the bottom, and what's left in the so-called supernatant, which is the liquid above, would be whatever sort of shook off or did not really get into the cell. And what their reasoning was the following. If the genetic material in fact is protein, then in the strain that has the radioactive sulfur, that will end up in the pellet. The pellet just means when you centrifuge something, the part that ends up at the bottom. It's the cells in this case. So in other words, if that pellet is radioactive, when you use the sulfur labeled strain, that implies that it was the protein that got inside of the cell. Conversely, if DNA is the genetic material, the P32 labeled strain will, which is labeling the DNA, the radiation will end up in the pellet in that case. So what actually happened? I'm sure you can guess. Here's sort of a flow chart of what they did. So in this case, drawing it as if, so the DNA here is labeled in purple, and the coat, just to keep track of the coat, is labeled in yellow. Okay, so obviously they couldn't see what was literally going on here, but I'm showing you a picture of what's really happening, that the DNA is getting injected into the cell. And after you put it into the blender, the supernatant we made yellow because that has the viral code and all of the rest of the viral proteins in it. The DNA ended up in the pellet down at the bottom, right? So what you literally do is sort of pour off the supernatant, so you separate it from the pellet and ask, where's the radiation? Okay, so I think you can see that what I want you to get out of this picture, which is very important, is that in fact what happens, again, they didn't know this, is that until they did the experiment, is that when you do this experiment, the viral coats end up in the supernatant and the DNA ends up in the pellet. That's factually what happens. But say you did the experiment, you're looking for radiation. If you found radiation over here, that would imply that the protein did not get into the cell and the DNA did. If you found the radiation, I'm sorry, it does depend on which strain you're using, right? So let me just step back for 1 second, because this is an important thing to understand. I'm going to erase all this stuff right now. There's a slide in a minute that explains it a little better, but what I just want to be very clear. Say you just did the experiment and you used the radioactive sulfur. We know that it can happen, but if protein was the genetic material, in that case, the radiation would be found in the pellet, right? Because it would have implied that the protein somehow went into the pellet, and it wouldn't be found in the supernatant. I wrote viral coats here, but that's because we know that's what happens. Now, if, as is in fact the case, you use the phosphorus strain that was labeled radioactively, then you looked for where the radiation was. If you found it there, that would imply that the DNA got into the cells, and that's exactly what happened. But if you found it there, that would imply the DNA was outside the cell. I'm sorry if there was any confusion about that. Let's look at this picture, and I think if there is any confusion, it'll clear it up for sure. So what we did in this picture was we drew in red the radioactive part. You can see that these are side-by-side experiments. So firstly, on the one side, we had phages that were grown on the radioactive phosphorus, okay, labeled with P32, and that means the DNA is labeled, and you see that there because the DNA is colored red. Over here, you have the viruses that were grown on the radioactive sulfur, which means that the proteins are red, which is why we made the coat red in that case. So the idea is, since in fact this is what's happening, in other words, the viruses are injecting the DNA, but let's keep track of the radiation. So when they did this using the DNA labeled strain, the DNA was in the cell, and they found out that the radioactivity existed inside the bacterial cells in the pellet and not in the supernatant, okay? Because the viruses popped it right in there. In the other strain, they found out that we drew the DNA in blue here just to say that it's cool, it's not hot, it's not radioactive. The protein never went in, so after they put it in the blender, this ended up in the supernatant, and you saw the radioactivity within the viral coats in the supernatant, right? So if you put these results together, the only conclusion you can draw is that when the DNA is labeled that radioactivity ends up in the cell. When the protein's labeled, that radioactivity ends up outside the cell. So DNA is the information that the virus is putting in. Thus, DNA is the genetic material. That's the deal. So after all of this occurred and people had time to process it and think about it, it was accepted that DNA was in fact the genetic material. And in the early 50s, people got very excited about this, as you can guess. And it really started to be a race coming on to figure out. Now, for so long, all the attention had been given to proteins, if you want to think of it that way. Now, DNA was starting to get some attention. So we had to start asking ourselves, what's up with DNA? What's known about it? How could it fulfill the conditions we mentioned earlier? Information storage, ability to replicate itself, and capacity for variation. So the question that next needs to be asked, and which we'll address in the next session, is at that time, what in fact was, from a biochemical structural point of view, known about DNA? Since we pretty much now established this is this thing, this is the genetic material. Now we have to really understand how DNA can be the genetic material. What is its structure and how does it relate to the function? And that'll be the topic of the next session. I'll see you then.